So hello and welcome to our webinar about temperature control in high resolution microscopy. I am your host. My name is Daniel Böning. I'm representing the company Interherence, and I will guide you through this webinar. Concerning the content of this webinar will be like the following. So I will give a brief introduction to our progress in temperature control concerning high resolution microscopy. And then we will have two speakers. One is Mart Krupovic, and the second one is Philipp Tripal, who then will give a scientific talk about their actual research and temperature related issues in their daily life. You can ask questions by writing your questions in the chat. And you can also raise your hand, and then I will unmute you, and you can also ask questions directly to the presenting guy after his talk. Questions related to my talk will be answered at the end of this webinar. And one more thing is that this webinar will be recorded, and it will be available via the same link, the Interherence webinar.interherence.com link you got for the invitation. So you can look up the webinar later on. So my part is now to show you kind of the motivation how we got into temperature control in microscopy. And this is our team, the Interherence company, consisting of 7 members. And our origin was mainly the Max Planck Institute for the Science of Light, where we basically developed new methods for super resolution microscopy and on-chip single molecule detections. And one particular thing is temperature control. And if you look at the temperature control systems which are out there in labs, it's a huge incubation chamber built around a commercially available microscope, and then there's a heater which supplies the incubation chamber with hot air to achieve a certain temperature inside the incubation chamber. And this system has some issues or drawbacks, or which I would like to mention now. So first of all, the chamber is very huge, so this makes the reaction time very long concerning temperature changes in your volume, in your sample. You have low accessibility, meaning that you cannot put many devices inside your incubation chamber. You have a restricted temperature range because you're limited to the power of your heater element, and your optics are sensitive to elevated temperatures. And one more thing is that if you want to use immersion objectives, there is a heat flux between your sample and your heated chamber and your microscope objective. And this is shown in this image. So basically your cover slip is passively heated by your heated air, and the immersion oil thermically connects your objective with the cover slip, and this leads to not equal temperatures on top of your microscope objective. In fact, in the center of your field of view, you might have a much lower temperature than the actual adjusted temperature in your incubation chamber. And due to this, your objective, because it's connected to the table itself and to the rest of, via the rest of the microscope, it's acting as a heat sink, such that it results in uneven temperature distributions in your field of view. And this is basically our motivation. So we wanted to make these devices much smaller. We wanted to make temperature changes much faster. We wanted to make it more precise than this type of arrangement, because in these type of arrangements, you might have a temperature sensor which is far away from the sample, and this might lead to not very accurate temperature measurements in your sample. We would like to make optics not being affected by temperature variations, and we would like to make it easily accessible, such that you can put more devices inside or near your microscope to allow for additional experiments. And we would like to make it cheaper. And our solution to that, or our idea, is we have a glass substrate, which is basically a conventional glass cover slide, which you also use for high resolution microscopy with a thickness of 170 micron, which is quite common in this type of microscopy. And this glass substrate has an optically transparent heating element in the center, and the temperature sensor, which allows to make use of a feedback system between these temperature sensors and the transparent heating element, such that the temperature can be adjusted in your field of view. This is actually the bluish area where you want to measure, can be adjusted very precisely. And these substrates, they can be fit into a substrate holder, and this is here shown as a microscope adapter, which fits in all commercially available microscope slide holders for these thicker and elongated glass slides, such that this system is compatible in its closed form, where you want to measure with all commercially available microscopes. And together with our control unit, we are able to ensure to have a well-defined and homogeneous temperature in the field of view. This allows us to being fast and precise in terms of temperature dynamics. We can go up to very elevated temperatures with up to 300 degrees Celsius. It's a plug-and-play system, which allows you to install the device very quickly and start your measurement. And it also minimizes aberration in optics, in contrast to the commercially available other incubation chambers a lot. And this is shown on the next slide. So we measured the performance of our system by measuring the point split function of small particles placed on top of our substrate, and the geometry of this point split function gives us an insight where how our optics are affected by the temperature elevation. And for the for air objectives, we basically see no difference in the size of the point split function with elevated temperatures. And for the immersion oil objective, we see a 30% increase in the point split function size up to temperatures to 80 degrees Celsius. And upon that, we can also, I would like to show you the temperature dynamics of this system. So this is shown on the left side. We have a substrate, and then we, in blue, the blue curve represents the set point of the temperature, and the red curve is actually the actual measured temperature on top of the sample, and with no additional substrate on top, we measured this red curve, which basically fits very much to the set point value, so we can achieve heating rates up to 100 degrees Celsius a second in this case. And since the system is not cooled, the cooling rate is dependent on your room temperature only. And this can also be done with liquids. So to hold liquids on top of our substrates, we have a reservoir made of PDMS, which is quite common in microfluidics. So it's very compatible with cell experiments. And also in this case, we can show that the measured temperature pretty much follows the set point, and we achieve very high heating rates in this case. So you can change your temperature between room temperature and 37 degrees in this case within a liquid environment within less than a second. And this allows for some kind of interesting experiments. So one thing is to apply heat shocks to cells. So this is shown in the video on the left side, where the cells are exposed short heat shocks, and then you can see that after giving them too much heat, they just die. So the first heat shock is applied with 45 degrees Celsius, and the second one is 55, and after that they are done. Additionally, our device also allows for very precise and temperature adjustment and therefore long-term stability temperature adjustment, which is shown in these graphs. So this is an overview of the temperature. The VAHEAT device was switched on at this point, and then the measured temperature again follows the set point value. And on the right side, there's a zoom in of this curve, showing that the precision by which you can actually hold the temperature is less than 0.1, much less than 0.1 degrees Celsius in this case. And this leads to experiments which can be performed over hours. So one particular example is to measure cells for a very long time. So on the upper right corner, you see that it's a few hours where cells survive at 37 degrees and they move, and the image you see on is showing fluorescence of tubulin, and so this results in another possibility of the device to allow for very long measurements. And the last thing we tried together with the Optical Imaging Centre here in Erlangen is we also imaged neurons and imaged them at temperatures around 37 degrees Celsius, and try to estimate whether there's temperature dependence in transport velocities of vesicles inside these cells. So, and this will lead to the talk of Philipp Tripal, which will talk right after me. This brings me to the end of my short introduction. So I will introduce the speakers now. So the first speaker will be Philipp Tripal. He is a scientist at the Optical Imaging Centre in Erlangen. He is an expert in various optical high and super resolution microscopy techniques such as confocal, Turban, palm storm, and that's mainly their application in live cell experiments. So he's a biologist, and in his talk he will use some of these techniques to study temperature-dependent transport processes in live cells, especially in neurons. And our second speaker is Mart Krupovic. He's the head of archaeal biology at the Institut Pasteur in Paris. His research is focused on understanding the morphological and structural diversity of archaeal viruses and uncover some of the molecular adaptations underlying the stability of archaeal viruses in extremely hot acidic environments. In his talk, he will highlight the mechanisms of virus-host interactions in Archaea, especially those which could be further understood through development of live cell imaging approaches. I saw some of your questions in chat, so I will answer these questions at the end of the webinar to give the speakers now the chance to present their research. And yeah, please keep your questions, and I will answer them later on. So now I will let Philipp talk. Now Philipp can, in principle.
Yes. I have to share my screen. You have to stop sharing. Okay, now I can share.
So Philipp, the stage is yours. I will unmute myself and you can continue with your talk.
Thank you. You see my screen?
I see your screen, yes.
Okay. So thank you, Daniel, for your kind introduction. And of course, thank you for the chance to be part of this webinar. As Daniel already mentioned, I'm a cell biologist in the core unit for optical imaging. So every day I'm confronted with different imaging tasks, and most of them are live cell and go deep and high resolution. And with my today's talk, with my presentation, Biologic Requirements in Live Cell Microscopy, I want to point out important physical factors which have to be controlled or at least monitored when it comes to live cell microscopy. The first thing, when I went into the optical imaging, I had to learn that it's not all about biology. There are really important physical factors to control, and we cannot have it all at the same time. So, of course, I would love to have high resolution, speed and light. This is impossible. We have to focus on one thing, and this is what I already recommend to you before you start planning your experiment or your live cell experiment. Make sure you find out what is the most important factor for you, so you can focus on that and then really get high quality in this one factor. You cannot have all at the same time. As a cell biologist, there's, beside these physical factors, resolution, speed and light, there's another really important factor for me, is the survival of my sample. I mean, it doesn't make sense to put 2 million euros in an instrument when your sample is dying. So this is what is a really important factor for cell biologists is the survival of the cells, and the survival is correlated to resolution, speed and light, as I hope you will see during my talk. There's one big issue: how can we monitor cellular viability, and how can we prove on the instrument that our cells that we show in the research are happy, or like the cells are doing good? The most important thing is that the control and the sample have the same conditions. So it doesn't have to be the optimal condition, it just have to be the same conditions for good research. Of course, it would be a pleasure to have the best cellular conditions, but this will, as you will see in my talk, this is really a challenge. So what are the physical factors impacting cellular viability in live cell imaging approaches on the microscope for the researcher? The first thing you have to think about is humidity. Humidity in the incubator. In incubated cell microscopes is not a problem. You just have some water either in the incubator or in the host of the oxygenation, or like the carbon dioxide host. You can just control the humidity. The humidity is something, there's no really monitoring system for that. So there's to monitor humidity with fluorescence or imaging based is really hard. it's still a really important factor, especially when we image neurons and you have evaporation in your sample, you will have different results. A next really important factor for me as a cell biologist is sterility. As I already mentioned, we don't need a 2 million instrument when our cells are dying or suffering. I do all my research in the absence of antibiotics, so I quickly see, and like contamination is something on microscopes that you really see when it happens, except this mycoplasma, and this is what you can test with some other test kits. But this is also something I really think is an important factor when it comes to cell microscopy, live cell microscopy. Another factor, and this is something that Daniel was already referring to, is temperature. Temperature, for me, I thought this is easy. I can incubate my whole system. I heat up the instrument 3 hours before I start the imaging, and then I have stable conditions. I will refer to that. I have some data which I was really astonished, and I never thought that there might be such a bias, such a temperature bias that I cannot really control in normal incubated imaging systems. Another factor which is really important is photo bleaching. Of course, photo bleaching is not directly affecting the cellular viability, but photo bleaching causes that I have to increase either exposure time or laser intensity, and this in the second step will have an effect on cellular viability. So it's really good to have these new dyes which are coming up, which almost show no bleaching, and there's also fluorescent proteins with really nice stability and high intensity, which can be used. I show you some data about this. This is something you have to worry about when it comes to live cell imaging approaches. The carbon dioxide concentration in the sample, of course, this is a really important factor. You can omit this when you work in HEPAs buffered systems, because the carbon dioxide changes the pH level in the medium. Everybody knows, we see that in buffered media when there's a shift in the phenol red in the color, then you can already see that there's something happening. I show you also some techniques how you can monitor and control this carbon dioxide concentration in your live imaging approach. And the most important thing, of course, is phototoxicity. And this is something there's really a big debate about it, how to control that, how to monitor that. I give you a really, what I think a really nice solution for that. It's not easy to perform. And over the time, there will be quality standards probably which will deal with that point. But at the moment, this is, for me, it's one of the most important things you have to worry about when it comes to live cell imaging approaches. Another really important thing I will show you during my talk. This is something I never expected. How can we define the perfect condition? This is something that is also not really simple to do. Probably you have to define it according to your result. If the carbon dioxide concentration is important for your research, you have to focus on that. Or if phototoxicity is an issue, you have to focus on that. But how can you define the perfect condition? This is really elusive at the moment. As I say, the most important thing is that you have the same condition for your control as you have for your sample. And already this is not easy. I show you now some techniques how you can control these physical factors and how you can also monitor that. I just collected some papers. So for the oxygenation, there are fluorescent probes where you can directly measure in the sample. By fluorescence, you can measure the oxygen concentration in the liquid. There is the same thing for nitric oxide. You can also measure that by fluorescence. You can measure the pH by fluorescence. You can even measure the ATP to ADP ratio. And as I already mentioned, you can measure with fluorescence the concentration of carbon dioxide in your sample. This gives you the chance to prove the concentration in your sample at the time of imaging, and then you can also show that it's in the control in the sample. You have the same settings, and this is good science. Just to show you how this works, so to measure the oxygen concentration with fluorescence, there's, for example, there's a foil you can put on the cover slip. This foil contains fluorescent molecules which change their fluorescence according to the oxygenation. And when you see now this oxygenation curve of a sample cells on the sensor, so number A, there you see that if the cells cover the sensor, you have a fast decrease in the oxygenation in the sample. If the cells sit beside the sensor in B, then you have a slower reduction of the oxygenation in the sample. So this clearly proves that we are able to control oxygenation by fluorescence. But how can we monitor cellular viability? There's a really nice publication from Spencer Short's lab where they show, and this is also something I saw, that if the cells are suffering, they reduce their cell division. So the amount of cell division is a good monitor for cellular viability. So there's a nice protocol from Spencer Short's lab. First, you choose a microscope, as I mentioned. Then you choose the exposure time. You keep this constant. You select an illumination power, keep that constant. You prepare embryos at a pronucleic stage. This is something that is probably not so easy for most of the people, but this is how you do it. Then you stain for the nuclei. You wait for the first anaphase to appear. Then you image this for 2 hours in 4 dimensions, so a C-stack over time. And then you count the number of nuclei. And you can calculate, if you compare the embryo from the incubator with the embryo in the microscope, if they have the same number of nuclei, then you know there's no effect of the microscope on our cellular viability. If there's a difference, then you already know, oof, we impact our cellular viability by our microscopic settings. This is really not easy to perform, but this is what I would say as the gold standard for to test for cellular viability. I choose this system here, as you already saw from Daniel's talk, is a full incubated spinning disk system with a high sensitive camera. I have long working distance lenses, all with immersion. I can adjust hypo and normoxy conditions. And with this instrument, I can control CO2, O2, and temperature. And it's no problem to do long-term imaging for more than 24 hours. This is something I will just show you. I'm able to do long-term live cell imaging in fluorescent small intestinal mouse organoids. So these organoids are really sensitive. But as you see, if I play this video, I can image that for more than 40 hours. And now you see it's playing slow. You see this one cell is getting bigger. You see it's doing a division. And then, so we are already able to record divisions in huge organisms. And from this, I recorded a C-stack. So we are also able to do three-dimensional imaging. Here, another example of a primary normal dermal fibroblast. This is transient transfected with a M-scarlet fusion product to a mitochondrial fluorophore. I recorded 4 tiles with 16 slices, so a C-stack of 4.8 micrometer with a 5-minute interval of 772 time points. And what you see is a maximum intensity projection. If I play that, you will see, of course, there is photo bleaching happening. This is, I cannot avoid this, but what happens also, there's another cell coming in. This cell is after 25 hours is starting to do a division in the field of view. So this is already showing me that my conditions in this approach are not so bad. Seeing divisions in the field of view is already a marker for good cellular viability. Once again, an organoid. Here you see the bottom of it. Here you see the center of the organoid. So this is a structure that is probably 100 microns thick. If I reconstruct the C-stack of that, you see it's proliferating. There is a new stem cell center forming. This all happens within 40-45 hours. And this was just done by conventional microscopic setups without the VAHEAT chamber. Now that we have this VAHEAT chamber available, and I had the chance to teach a student, a molecular med student, Pranjali Divika, and she was playing around with the VAHEAT chamber, and we tried to find out if we really have a high loss of temperature when we use this in the live cell microscope. So this is actually our setup, the VAHEAT chamber in a turf instrument, and I work with immersion. So we expect that we lose a lot of temperature through the oil, but this is something we wanted to test. And of course, I have to apologize, we don't have perfect instruments for a high resolution imaging of this, but on the left side, you see the microscope with the VAHEAT chamber and the attachment of the lens, and on the right side you see an infrared image where we are able to visualize the temperature in the center of our slide, so really in the point of view. If I play this now, you will see on the right side you will see the temperature. As soon as we touch the lens, the temperature is going down. This instrument was pre-incubated for 37 degrees, 3 hours. So this is actually what I thought is enough time to have a nice distribution. If we remove the lens from the sample, you see the temperature is coming up. As soon as we touch it, we lose the temperature again. And this was really astonishing. We lose up to 5 degrees in the field of view as soon as we touch the lens on the sample. Even so, this instrument was preheated, the whole incubator was preheated for 3 hours. So we now play around and test this if we pre-incubate longer, but this was really astonishing for us. So, and this is why I wanted to test if we already have an effect on the track of the speed of lysosomes within primary red neurons. For this, we choose TURF, the TURF instrument. This is not incubated, but we used the setup with the VAHEAT chamber. We were able to have nice primary neurons from rats in a cooperation from the psychiatry. We were staining the lysosomes. This is pretty easy with lysotracker. And then Pranjali was just in her 6 weeks practical. She was playing around and she was measuring the speed, or she was just tracking lysosomes at different temperatures, let's say like this. And at 30 degrees, of course, we see not much movement within the axons or the dendrites. It's some movement in the cell body. At 37 degrees, I play this again. Yeah, starts again. You see movement, the regular movement of the lysosomes within the axon. And if we increase the speed, the lysosomes are moving faster. This is now at different magnifications, and I'm sorry about that, but you can already see that there's much more movement. They start not move into one direction, they probably also start wobble around. And this was already, we think, the difference of 11 degrees, we see a lot of difference in the movement. In the next step, she now narrowed down the range. We image from 37 to 40 degrees, so just in a range of 3 degrees. And already 3 degrees make a big difference. Like when you see here, regular movement 37, at 39 it's speeding up and wobbling around. And at 40 degrees, of course, because of the drop in ATP, there's no more movement of the lysosomes. So this is, as I would say, preliminary data, but of course we see effect of temperature. So taken together, I can say we have physical factors that impact cellular viability and live cell microscopy. I show you there are ways to control and monitor these physical factors within the field of view. We have a variation of temperature within the field of view. as an effect, and this has an effect on the speed of lysosomes within primary neurons, as I show you, even in a narrow temperature range. And as Daniel was showing, there's a new tool available to regulate and monitor temperature directly within the field of view, which was one of the factors which was hard to assess directly in the position where you image. But now with this tool, we are able to do, and with this tool, I was really astonished that there's so much impact. With that, I like to end, and I love to get your questions after Mart talks. Thanks.
So thank you very much, Philipp, for your nice talk. Are there questions? So you can ask questions in the chat. Are there questions especially related to Philipp's talk? So I see only questions regarding the VAHEAT system itself, but are there questions related to Philipp's talk especially? So as people might think about questions, I will ask questions. So what do you think in a taking the existing heating systems and measuring at slightly different temperatures than 37 degrees, will it actually harm the cells very much? So if your temperature is slightly lower, like if you measure at 37 degrees, or are these measurements, so basically, is it harmful for cells if you measure them at 35 degrees?
So temperatures below 37 is well tolerated in nature, as we know. We can also put our hand in the winter and it's still alive. But above 37, this is really challenging. This is where we see that is immediately you have an impact on the cellular viability and all moving processes. And as soon as you heat it up higher than 39, this is already the border. Below 37, there's probably not much killing effects, but as I mentioned, you will have a bias if you do not control or monitor the temperature. You will have a bias between the sample and your control already, because as we see that the touching of the lens reduces a lot of the energy within your sample, you will never be able to prove that it's the same effect in your sample that you have in your sample and you have in your control. And this is why I was really, I never thought about it. I thought I heat that up, it's 37, and then I don't have to worry about this anymore. But as we see with the infrared camera, there is an enormous effect. This is
okay. So one short question which came up via chat regarding your talk. So could you just in a few sentences, in 1 or 2 minutes, describe how you put the samples with the liquid media on these cover slips, on the VAHEAT cover slips themselves? So how did you do that?
This is something, of course, this took some time to establish, especially for the primary neurons. They are sensitive cells and you have to play around with the coating and stuff. But usually it's really easy because the cover slips, the VAHEAT cover slips are already with a chamber. So we have a media chamber sitting on the glass, on the cover slip, as you already saw also from the images. And you have a volume of roughly 400, 500 microliter. You can put media on that. Of course, you have to worry about evaporation because it's not so much media, but if you have a full incubated system, the humidity should be good enough to prevent evaporation. And then, of course, you have to play around with the coating. Some cells don't like the glass surface, but you coat it with polylysin, like a amino acid, and this is how we made the neurons adhere on the glass. This is polylysin, or there's also poly-L-ornithine. There's many substrates you can test, and for each cell type, you have to play around and find out what is the best. Yeah.
Okay. Thank you very much, Philipp. Then we hand over to Mart.
Should I share my screen now?
Yes. you can share your screen. Philipp will mute himself, right? And I will also mute. So I'm very happy to. Well, I'm looking forward to your talk. Thank you for having you with us here, and yeah, I'm very excited.
Okay. So first of all, thanks Daniel and Pierre for the invitation. I'm always happy to share some of our research and my fascination with hyperthermophiles. So my talk will be a little bit different from Philipp's in that we actually don't have much data on live cell imaging, but that is something we want to achieve. And temperature, it is clear that it's very important for us because we are studying organisms which live in these kinds of environments. So on this slide, you see one of the hot springs which we have sampled. And generally, when someone sees this kind of hot spring, I think an immediate question is whether life can exist in this kind of setting, because it's not only hot, but also very acidic. So it's more or less boiling acid there. And one thing we know for sure is that human beings definitely cannot survive there. And occasionally there are, accidents, or some people do it willingly, like in the case of the report in The Guardian a few years ago. So they try to get into the hot spring, but generally this ends bad, and human bodies are never recovered from these kinds of hot springs because we simply dissolve. But microbes, they can live there. And this is particularly true for Archaea, which I think you all know are members of the third domain of life beside bacteria and eukaryotes. So in these kinds of hot springs, Archaea dominate. And it's not that they can survive in these conditions, but they actually flourish there and prosper. And here you see a graph of the growth dependence on the temperature. And our lab model is Sulfolobus, and you can see that optimal growth temperature is around 80 degrees. And if the temperature is low, we simply do not want to grow there. So we need the temperature. And some archaea, such as in the case of Pyrolobus, we grow optimally at temperatures above 100 degrees. And if you offer something like 90, no, we don't grow. And these bugs, they live in hydrothermal vents. So temperature in our research is of essence, of course. And there are many different kind of hot environments on our planet where there are microbes, and we are studying these hot springs, mud holes, geysers, deep sea hydrothermal vents. And in my lab, we are focusing less on the microbes, but rather on the viruses which infect these microbes. And it turned out that the virosphere associated with hyperthermophiles is astonishingly diverse, right? And here you see a kind of a collage of different EM pictures of archaeal viruses. So you see typical viruses with icosahedral capsids, which you also see in bacteria and eukaryotes, but the vast majority of these viruses, they do not resemble bacterial or eukaryotic viruses. So they are unique to Archaea, and most of them are unique to hyperthermophilic Archaea, right? And here you see some kind of iconic shapes, such as the champagne bottle-shaped virus or lemon-shaped virus, which you don't see anywhere else, right? And in the case of this, for instance, lemon-shaped virus, these are produced by the cell as baby lemons without the tails, but then once outside of the cell, they undergo morphological transition, and specifically they grow tails at both ends of the particle, right? And eventually in the population you have almost all viral particles with the long tails at the ends, right? And the only requirement for this transition is actually hot temperature. So if you incubate tailless virion at room temperature, nothing happens for a year. You can keep it, it will stay the same. But once you put it at hot temperature, it will undergo this transition. And what we are interested in and what we are working on is mainly three questions. So first one is the extent of archaeal virus diversity. Second, how these viruses remain stable in very extreme environments, hot and acidic environments. And third, how these viruses interact with the host cells, right? And there, perhaps the live cell imaging is most relevant to understand the last part. Okay, next, so I promised to tell reveal some of the secrets of archaeal viruses, how they manage to survive in these conditions. And some of the insights we obtained from structural studies of the viral particles. So this is the rod-shaped virus, SIRF2. It infects sulfolobus cells, which grow optimally at around 80 degrees, and pH is 2-3, so hot acid. Right? And in collaboration with Edward Egelman's group, we obtained the structure of this virus. And it turned out that the capsid protein is fairly simple molecule. So here you see a dimer of a major capsid protein, homodimer, and it has a 4-helix bundle with a long arm attached to it. And it's a homodimer which forms this claw-like shape. And as all claws, what it does, it grabs onto double-stranded DNA, a linear double-stranded DNA of the virus, and completely insulates it from the environment. And this is of utmost importance in the acidic environment, not to allow the acid to get in contact with the viral genetic information. Right? But in doing so, the kind of tightly wrapping around the DNA, and this goes along the entire genome. Each base pair is complexed with a protein. And what this leads to is dehydration of the DNA. And we were quite astonished to find out that the normal physiologically common B-form DNA in the viral particle is actually transformed into the A-form DNA. And this conformation of DNA has been discovered many years ago by Rosalind Franklin, and it was assumed ever since to be an artifact, a laboratory artifact due to dehydration of DNA. But now we know that this form of DNA is actually biologically relevant, and it is found in archaeal viruses. And I was telling about one virus, but we now know that many of archaeal viruses, rod-shaped and icosahedral also, they store the DNA in the A-form conformation in the viral particle. Okay, so another thing which we are interested in, as I mentioned, is how these viruses interact with the host. And again, the same virus, and it recognizes the host cell, sulfolobus islandicus, by binding to the type 4 pili, which are abundantly present on the host cell surface. And the virus binds to the pilus at the tip, and we can obtain this binding in vitro, isolate pili, mix it with the virus, and it will bind to the tip of the pilus. But this binding, it will happen only at physiologically relevant temperatures. So at 80 degrees it will bind, at room temperature there will be no binding. So it's a temperature-dependent interaction between the host and the virus. So the virus binds to the pilus, and then it somehow runs along the pilus to the cell surface, right? And this happens in 1 minute. The pili can be up to 12 microns long. The virus is around 1 micron, so 12 times longer pilus, and then virus binds to the tip and then runs along. And this happens within 1 minute after virus addition to the cells, right? So extremely fast process. But when the virus takes around 10 hours to do things inside the cell, to assemble new virions, which you can see here, and then they have to be released. And this virus does it in a remarkable way. So it builds virus-associated pyramids on the cell surface, which you can see here in the thin section of the infected cell, these large triangular things right at the cell membrane. And here you can see in scanning electron microscopy. And these actually are pyramidal structures. So they are formed from 7 triangular plates, which form the 7-sided pyramid. And what happens when the time is right, when the virus has to be released, these pyramids open up pretty much like petals of the flower opening up and forming holes in the viral, in the cellular envelope through which the virus can be then released. And here in scanning electron micrographs, you can see kind of cells with many holes. And eventually what you are left with is an empty shell where the virions are gone, the cellular contents are gone, so just a shell, dead shell of the cell. Now, not all archaeal viruses are as deadly. For instance, in the case of a lemon-shaped virus of the Fuselloviridae family, which infects very closely related hosts, Sulfolobus, again, hot and acidic, the virus is assembled and released by a very different mechanism. So here you see the electron tomography kind of volume where the virus is being produced by extrusion of the nucleocapsid or nucleoprotein filament, which you see in red, through the cytoplasmic membrane of the host, right? And then the particle is formed and extrudes through the surface layer, which covers the archaeal cells. So it's just a protein layer on the outside. In the next step, this budding particle is being constricted at the base, and eventually the membrane scission takes place, and the particle departs into the environment. And this type of virion assembly and release, they are very much similar to the budding of different enveloped eukaryotic viruses, such as HIV, influenza, and many others. And the last bit of kind of on virus-host interaction is another system which consists of the yet a different virus, also lemon-shaped, but belongs to a different family, much larger than the previous one, and it has one tail at one of the pointed ends. So the virus also infects Sulfolobus cells. And Sulfolobus cells are around 1 micron in diameter, spherical, as you can see here. But what happens with the cell when the virus infects it is that it grows in diameter immensely. So in 3 days, we see that the cells become some 6-8 times bigger than the non-infected cells, right? And so we wondered what happens if you wait longer. Well, you wait longer, you get much bigger cells, right? So in few days, we have observed up to 20 micron diameter cells, and this is huge compared to 1 micron. To get to 20 micron, which is around the size of a kind of many eukaryotic cells. And here you see the infected cells in scanning electron microscopy, so non-infected cells. And here on this image you see non-infected cells, normal infected cell, but still of a normal size, then considerably bigger cell, and then a much bigger cell, right? And the virions are here kind of sprinkled at the surface of the cell. So this increase in the diameter of 20 times means that the volume increases of the cell by 8,000 times, right? And this is remarkable. And here we can see all the transitions between the kind of tiny normal-sized cell of 1 micron to something way bigger. And these cells contain up to 300 copy equivalents of the genome, whereas the normal size, they have like 1 or 2 copies of a chromosome. So they are filled with DNA, filled with content, huge. And I think we should all feel lucky that our viruses are not doing the same to us. Otherwise, kind of, it would be really crowded very quickly in the times of pandemics as we have now. Right, so thus far we have been studying these archaeal viruses and their hosts using mainly electron microscopy and electron tomography. And this means that we are looking at the still images or snapshots of different time points after infection. And of course, the kind of the goal which we want to achieve is to be able to observe the infection process and different processes which happen in the cell during the infection in real time. For instance, how fast or how this virus moves along the pilus when it infects in a rapid process, or the envelopment of the viruses inside the cell. And we know that for some viruses, this this is happening inside the cell. We somehow acquire lipids very selectively, but we have no idea how this is happening. And how these, beautiful flowers, the pyramids, how they open, and how quick this process is. And the budding of the viruses, and also the gigantism. How does the cell grow, so big, and how does this thing happen? So for all that, live cell imaging would be really something very, very useful. And a big challenge, of course, for a long time with working with hyperthermophiles was the temperature control. And for obvious reasons, because we have to offer around 80 degrees to the cells to do anything there. And with the VAHEAT system, it seems that we might have a solution, and we are currently testing the system, and it shows a great promise. So we might have this part of the difficulty solved. But we still have other challenges. For instance, all the endogenous fluorescence markers which you are used to use, like GFP and Cherry and many others, they simply not available for hyperthermophiles. So these proteins would get denatured immediately, and there is no fluorescence, so we cannot use that. Also, a similar thing with the fluorescent dyes, which are commonly used. They are either not thermostable, they don't fluoresce at high temperatures, or they are cytotoxic. So again, we really have a very few possibilities with getting any kind of fluorescent labeling for the live cell imaging. So we are still a long way to go to be efficiently using live cell imaging, but we are making good steps forward. And I would like to thank many people whose data I presented here, and we are working with people at Pasteur to develop the live cell imaging using the VAHEAT system for hyperthermophiles. And thanks. I'm happy to take any questions if you have any.
Thank you very much, Mart, for the very, very interesting talk. So I have some questions. Probably people need in the chat need some time to formulate their questions. So I like to talk very much. So I have some questions regarding the.
Oh, sorry, I cannot hear you anymore.
Can you hear me now?
Very, very silent. It was okay first, but then it kind of dropped down.
Better?
Oh yes, no.
Okay. So I have a question concerning the microbes and the survival strategies of the host cells. So how do they do it? Because I did some cell experiments, and I always figured out that proteins denaturate at a certain temperature. So how do they protect, for example, their proteins for being denatured?
Yes, so of course there are adaptations in the proteomes of these hyperthermophiles, and that's one of the reasons why, like if you put GFP into this cell, it will get denatured naturally. But what is known, if you compare homologous proteins from mesophiles and hyperthermophiles, what you see is that for most of, that's a general adaptation for proteins, is that their hydrophobic cores are much larger in these thermophilic proteins. So more hydrophobic residues which hold the core together. We have much fewer loops. So all these floppy parts of the protein, they are gone. So we have really short loops. We have increased level of disulfide bonds in the protein. But we also know that almost all proteins in sulfolobus, for instance, are methylated, which probably also another adaptation to high temperatures. So like in eukaryotes, it's usually some proteins which are methylated, and that's for signaling. But here, any protein, it will be heavily methylated on the surface. Mm-hmm. Yeah, so these are main adaptations for proteins to sustain these kinds of conditions.
So, and my second question would be, concerning the markers to specifically label some parts of cells or virus of these viruses. There are of course some labels like inorganic labels like gold or so you could use as a marker instead of a fluorescent protein. However, you kind of have to attach these labels to your structure. What is the current research on that to kind of attach a label at these high temperatures?
Yeah, no one is doing that because, you know,
okay.
Imaging, right? So, or there was no live cell imaging. So like with the fixed cells, you can use antibodies and
yeah.
That's it. But for live cell imaging, attaching labels, yes, it would involve probably some genetic manipulation, which is also not trivial in these systems. So it's not E. coli, right? We have genetic systems, we can do some things, but not very sophisticated ones. Introducing tags or something like that. So that's one possibility.
Okay. Okay. Thank you very much. Yeah, for this nice talk. So, I'm asking the audience now, are there any questions related to Mart's or Philipp's talk? Please raise your hand or please ask them in chat if you don't want to. So I actually cannot see any more questions. I'm gonna check the chat. There are only questions regarding the VAHEAT device so far.
That's fantastic.
You can also have a double check on the chat, I think, but I don't see any more questions. So then, Mart, there's one last question from me. Is the cell size somehow limited in some case due to some factors? I don't know.
Well, at least in these cells which we are studying, so there are some other kinds of archaea, but in Sulfolobus there doesn't seem to be limit in size, so they can grow big. With of course normal cells, we control this size by dividing when they have to. But in the case of virus-infected cells, so you see they can grow up to 20 times larger, and that's fine for them.
Okay.
There is no peptidoglycan or things like that in Sulfolobus. So in bacteria, typically peptidoglycan restricts kind of growing cells in some directions, right?
Yeah.
In Sulfolobus, there is only membrane, which is surrounded by protein layer. And that's quite flexible, apparently.
Okay. Yeah. Okay, then thank you very much. I'm gonna reply to the questions which were asked in the chat. So the first one was asked by Sam Lord. So I can allow you to talk, actually, to reply to my answer. So you asked, for example, is the ITO coating on the bottom of the cover slip? Yes, it's on the bottom side towards the microscope objective. The sensor is actually on top of the substrate, and it's covered with a thin layer of glass. So the cells which are grown on the VAHEAT smart substrates are just seeing glass surface. And the second question would be of you was, if you use an oil immersion objective, do you still use an objective heater to reduce the heat sink? No, because we have a feedback system between our sensor system and our heating element. We actually measure the temperature in the field of view. And so that's why we don't need any additional heater. Sam, you can...
So presumably the area outside the field of view gets above the set point temperature.
One more time, once again.
Then presumably the area outside the field of view or not touching the objective probably goes above the set point temperature?
So the heating element is quite large, and the temperature is the sensors in the center of this heating element. And then if you have immersion oil, you very much distribute your heat load over the whole area, such that there are no big temperature gradients anymore in your field of view. So less than a degree, way less. Yeah.
Cool.
So did I answer all your questions? Yeah.
I believe so. Do you know, I have another question unless someone else does, but just quickly, do you know if these samples are compatible, the substrates are compatible with cleaning steps like plasma etching or chemical cleaning to make the sample, the substrate extra clean?
I can shortly answer this question, but then Philipp can continue probably. Just by the way, so Erdalat Radfa will be the next guy who can formulate his questions. He also asked something in the chat, so I will reply to your question after this one. So you can actually clean the substrates and reuse them. Sometimes you can clean them with all solvents like ethanol, acetone, isopropanol. You can also sterilize them in these solvents. You cannot sterilize the substrates in like in a sterilizer at 140 degrees with high gas pressure of water. But you can use solvents. You shouldn't use acids and yeah, to clean them, but solvents are possible.
Okay, thank you.
Okay. Okay. So now I gave the right to talk to me to Erdalat. Can you hear me, or do you want to talk? If not, I would just answer your questions. So you asked about the thermostability of all other parts, such as stage holders and so on and so forth, about the VAHEAT device, I assume. So in our device, for our device, only the substrates are heated. So actually not anything else. So all the other parts will remain at room temperature apart from the substrate itself. So that's why there is no thermostability needed. Your next question was answered by Philipp already, how to put samples with liquid media on these kind of cover slips. And then your next question was that I should explain the effect of this on photon counts and the image quality. So I can just talk about photon counts at elevated temperatures. If you use, for example, an oil immersion objective, so basically what you do with heating your substrate, you also heat your immersion liquid, and this results in a decreased numerical aperture of your objective in the end, which results in a decreased number of photons you can collect from your sample. And this I showed already in my talk. This also results in then larger point switch functions of what you observe. So yes, in principle, at some temperatures you collect less photons, but this effect is up to 80 degrees Celsius quite minimal. So we see an increase in the point switch function size in lateral. We don't see an increase in the point switch function size in lateral dimensions up to 80 degrees Celsius. We see an increase in the point switch function size in axial direction by about 30%. at temperatures at 80 degrees Celsius. So we collect less photons, but it's very limited. It's almost the same as if you would image at room temperature. Okay. So did I answer all your questions, Edalat? Okay, he's not responding anymore. Okay, so I think there are no more questions in the chat. There was a question concerning the price. I can tell you price if you ask for a quotation. So we can stay in contact, and you just drop me an email, or I reply via email to you with a quotation of our devices. That's how we do that. Yeah. So then I would like to thank all the speakers. Thank you, Mart. Thank you very much for joining the seminar. Thank you, Philip. Thank you both for giving us these insights in your research life. And yeah, hopefully see you soon, and have a good evening. Bye-bye.